So, when microorganisms were known to exist, most scientists believed that the simple life form could surely arise from spontaneous generation. That is to say, life was thought to spring spontaneously from mud and lake or anywhere with sufficient nutrient. This concept was so compelling that it persisted until late into 19th century. So the controversy of spontaneous and non-spontaneous generation were resolved by a series of scientists and their experiments, among which Reddy and Pasteur's work is the most prominent. So let us discuss them one by one. Reddy was an Italian physician who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously, and the first one to challenge the theory of spontaneous generation. He proved that life originated from complex form of life. So living thing originate only from living thing. He used jars containing meat which were covered and some were not. And the maggots only appeared in the uncovered jar. In Reddy's experiments, he used jars. Half of them were covered with wire gauze or sealed, or half of them were uncovered. His hypothesis was supported when maggots appeared only in the uncovered jar, but no maggots appear in the gauzed or tightly sealed jar. He concluded that maggots could only form when flies are allowed to lay eggs in the meat, and that maggots were the offspring of flies and not the product of spontaneous generation. The debate over spontaneous generation continued well into the 19th century, with scientists serving as proponents of both sides. To settle the debate, the Paris Academy of Sciences offered a prize for resolution of the problem. Louis Pasteur, a prominent French chemist who had been studying microbial fermentation and the cause of wine spoilage, accepted the challenge. Pasteur made a series of flasks with long twisted neck called swan neck flask, in which he boiled broth to sterilize it. His design allowed air inside the flask to be exchanged from the air from outside, but prevented the introduction of any airborne microorganism which could get into the twists and bends of flask's neck. If a life force behind besides the airborne microorganism were responsible for microbial growth within the sterilized flask, it would have access to the broth, whereas the microorganism would not. He correctly predicted that sterilized broth in a swan flask could remain sterilized as long as the neck remain intact. However, should the neck be broken, microorganisms would be introduced and contaminate the flask and allow microbial growth within the broth. Pasteur's set of experiments disproved the theory of spontaneous generation and earned him the prestigious Alhambert Prize from Paris Academy of Sciences. Lord Joseph Lister, a famous English surgeon, is known for his notable contribution towards aseptic techniques for prevention and cure of wound infection. Lister concluded that wound infection too were due to microorganisms. In 1867, he developed a system of antiseptic surgery designed to prevent microorganisms from entering wounds by application of phenol on surgical dressing, and at times it was sprayed over the surgical area. He also devised a method to destroy microorganisms in operation theatre. by spraying a fine mist of carbolic acid into air, thus producing an antiseptic environment. Therefore, Joseph Lister was the first to introduce aseptic techniques for control of microbes by the use of physical and chemical agents, which are still used today. Because of this contribution, Joseph Lister is known as the father of antiseptic surgery. Robert Koch in 1876 established that microbes can cause disease. He found that the blood of the cattle who were infected with anthrax always had a large number of bacillus anthrax. He found that he could transmit anthrax from one animal to another by taking a small sample of blood from infected animal and inject it into a healthy one. And this caused the healthy animal to become sick. He also found that he could grow the bacteria in a nutrient broth and then inject into a healthy animal and cause disease. Based on these experiments, he devised criteria for establishing a causal link between microbe and a disease, and these are known as Koch postulates. Although these postulates cannot be applied in all cases, they do retain historical importance to the development of scientific thought and are still being used today. So, according to Koch postulate, an organism to be classified as the causative agent of any disease, it must be present in every case of the disease. The agent must be isolated from the sample of the individual having the disease and can be cultured in lab vitro. So the disease must be reproduced when the pure culture of the agent is inoculated into a susceptible host. And eventually that agent must be recovered or isolated from the experimentally infected host. According to him, any organism which fulfills all these four basic criteria is classified as the causative agent of that specific disease. So, according to the postulate number one, the suspected pathogenic organism should be present in all cases of diseased animal and should not be present in any of the healthy individuals or animals. As you can see here, the suspected pathogen is only present in the diseased animal sample and is absent from the sample of a healthy animal. The second postulate is that the suspected organism should be able to grow in a pure culture in laboratory condition. As you can see, a plate showing colonies of suspected pathogens from the sample driven from the diseased animal. But these organisms should be absent from the sample of healthy individuals. The third postulate is the cells from the pure culture of the suspected organism should cause disease in healthy individuals. So that means the pure cultures from the petri plates can be used to inoculate a healthy animal which is susceptible to that pathogen, and it should cause the same disease, same signs and symptoms as in case of the original infected animal. Immunology is one of the two biologists which are credited for discovery of viruses. In 1890s, he was trying to find out the cause of tobacco mosaic virus disease, an infection that causes tobacco leaves to discolor. From affected leaves, he prepared a solution containing the infectious agent, and he passed it through a filter made of porcelain. This filter had pores so small that it can filter out bacteria. He found that after passaging through the filter, the solution was still fully capable of causing infection. This led to the discovery of the first viruses. In 1928, Fleming discovered penicillin, the first form of antibiotics. He grew cultures of bacteria on petri dishes in the hospital where he worked. A fungal spore happened to contaminate one of the bacterial culture and grew into a fungal colony. The bacteria around the fungal colony failed to grow, as well as the bacteria on the other sides of the petri dishes. From this, Fleming deduced that a substance produced by the fungus must be slowing down bacterial growth. He called this substance as penicillin after the fungus on the petri dish Penicillium notatum. Fleming's discovery earned him the Nobel Prize for Medicine. It led to the production of many antibiotics. Many illnesses such as lung and bladder infection, which are caused by bacteria, can now be cured thanks to antibiotic. So this table represents the highlights in the history of microbiology. These are the list of discoveries made in the golden age of microbiology.